Continuing on with protein tertiary structure, so far we have over 110,000 protein structures reported and documented. And from looking at all these structures, scientists have been able to get some general trends about amino acid locations in globular proteins. So nonpolar residues are in the center of a protein, and this makes sense because of the hydrophobic effect. Remember, water can't hydrogen bond with hydrophobic molecules, therefore they will push them and make them aggregate together, and that's what we see in proteins. While charged and polar residues are on the exterior. If you have a charged residue all by itself in the interior, that is very energetically unfavorable. It's possible to have pairs polar residues, for example an arginine and aspartic acid together, but just one by itself doesn't happen. Uncharged polar molecules, they're usually on the exterior, but if they are in the interior, they will be hydrogen bonding. And the interior of a protein is incredibly dense. There's not much space. It is well packed. Most of the water is gone. And it's like an oily substance, right? So you have all these hydrophobic residues all packed together on the inside. Also, from tertiary structures, we have seen that tertiary structures really can be just a combination of secondary structures. And so if we see secondary structures that are repeated over and over and over again, we call them super secondary structures, or more commonly motifs. So by looking at a lot of proteins, we have some common protein motifs that appear in nature over and over again. One common one being a beta alpha beta. And just by looking at this, you can see that motifs are just alpha helixes and beta sheets arranged. If you have three beta sheets together in tight turns, we call that a beta hairpin. If you have two alpha helixes that interact, that's an alpha-alpha motif. And if you have four beta strands, each connected by a linker, and then you have something going on where strand 1 and 2 are in a beta sheet, and 2 and 3, and 1 and 4, we call that a Greek key, because it kind of looks like this image, which is a Greek key. Now, talking about globular proteins and tertiary structure, depending on what type of secondary structure we see in the tertiary structure, you can label proteins as being primarily alpha, beta, or a combination being alpha beta. For example, in this protein, we have 4 alpha helixes, and you can see that in both notations, so we call this an alpha protein. This protein is just beta sheets, so it's a beta protein. And a sandwich like this, where you have beta sheets overlapping, we call that an immunoglobulin fold. You see it a lot in immunoglobulins. And if you have a mixture of alpha and beta, we call that an alpha-beta protein. For example, in this protein, 31% of the protein's in alpha and 28% is beta. Now, when we look at secondary structure like this, what we're really looking at is protein topology. So we're looking at how does the secondary structure look overall in the tertiary structure. What I mean by that is that we have a protein here that is nothing but beta sheets, but it forms a barrel. This is a 3D barrel. So the topology is of a beta barrel. Here we have beta sheets, and we have a lot of tight turns, so we call this topology that we're seeing, we say this is a beta hairpin. And you can also have alpha helixes in beta sheets as well, and we call this an alpha beta barrel. Here and in all the pictures we have seen so far, if you have an arrow, this means beta sheet. If you just have a rectangle, that means alpha helix. So you can see beta sheet, alpha helix, alpha helix in our alpha beta barrel. So continuing on with what have we learned by looking through all these structures, we've learned that large proteins, those greater than 200 residues, can fold into multiple parts called domains. Now I say parts, do realize that these are still connected amino acids. The peptide bond isn't broken here, but you have two very separate and distinct clusters that we call domains. Generally, a domain is going to be between 40 and 200 residues and 25 angstroms in diameters. And domains are usually structurally independent of one of each other. It's almost like you have two little proteins that just happen to be on the same protein chain. And when you start studying domains, what you see is that each domain usually has an independent function. One domain could be a catalyst for one reaction, and the other domain is a catalyst for a second reaction. Or a domain can just be a spacer for separate domains. Domains can have a lot of things, but they're usually independent of each other. And when we look at structure of different proteins in different organisms, we can see that evolutionary speaking, the thing that is conserved among organisms is how proteins look, not so much what amino acids in their sequence. So when we start looking at structures and domains and motifs, we see some domains popping up over and over and over again. And this is called an evolutionary sink. And so the idea is that these domains are very stable, and they're okay if some amino acids are mutated or not. So since these domains are very tolerant, it's possible that their sequence gets duplicated. You get a new protein with the same domain. That protein gets mutated, but continues to have that same looking domain over and over and over again. And a lot of these proteins, a lot of these evolutionary sinks are very important biological functions. And what we see is if we look at different organisms and we look at proteins that do the same job, their sequences can be very different, but they look the same. So here's cytochrome C in 4 different organisms. Now, if I just gave you the primary structure of these 4 proteins, you could not tell they were related. However, when looking at the pictures, you can see that they all pretty much look the same. So they are very much related. And the takeaway here is that generally, evolutionary speaking, it's more important that the structure is conserved than the actual individual sequence or individual amino acids. Now, what we've been talking about, looking at all these proteins, this field is called bioinformatics. And we have a place on the web where every single protein structure is stored, and this is called the Protein Data Bank, which is at this website. As of 9/15/19, there are 115,000 biomolecules in there, and that number just grows every single day. All this information has led to the field of bioinformatics or structural bioinformatics, where you look at large data sets and try to come up with patterns. Now, every one of these biomolecules is free to access, and they're stored in what's called a PDB file. So if you ever need to look at a PDB file or use one, this is what is inside a PDB file or protein data bank file. You have a line that says atom, that's telling you you're looking at an atom. You can have different names here, head atom. You generally ignore those and you just want to look at atom. What number the atom is, what is the atom's name, so hydrogen on carbon alpha, hydrogen on carbon beta, the second one, the third one, nitrogen. What residue we're looking at. What protein chain, if there's multiple chains, are usually called A, B, C, and D. What residue number, so cysteine is the 70th residue. It's X, Y, Z coordinates in space. How often you see this protein in the crystal structure? Usually it's always 1. Sometimes you can get something weird like 0.5. We don't need to worry about that. And temperature. How flexible is this residue in the structure? 99 means it is completely flexible and moves all around. 0 means it never moved whatsoever. And that is it for this video. I will see you in the next video.